All right, hello everyone. Welcome to the second part of the video lecture pertaining to the chemistry of life. So this is just going to end off with carbohydrates, lipids, as well as nucleic acids. So we're just going to finish up the chemistry of life unit. And I left off on proteins, so we're going to move on to carbohydrates next. So let's get into it. But actually, before we move on to carbohydrates, I forgot to mention that I also wanted to go over the concept of hydrophobic as well as hydrophilic, and as well as functional groups. As you can see, I have a big table there on the left. But again, moving towards the hydrophobic and hydrophilic concepts, the hydrophobic just means that a molecule is repelling water. And you can see the scatter water is right there underneath. But a good way to remember hydrophobic and its terminology in emphasizing that a molecule is fearing water, well, you see that it has that phobic term, right? Phobic, phobia just means fear in a way, right? So if you're contemplating between hydrophobic and hydrophobic just knows since it has that phobic, always remember that it just means the molecule fears water. It has that phobia of water in another way, in another sense of thinking of it. And then hydrophilic is just a total opposite. It means that molecule totally loves water. It loves to interact with water. So again, it just loves water. So hydrophobic means fearing water. Hydrophilic loves water. All right, now moving on to that big table to the left, as you can see, these are what are called functional groups. So we're going to discuss like the main ones. And starting off with hydroxyl group, it is defined by an oxygen binding to a hydrogen. And mainly you can find these hydroxyl groups associated with alcohols, and it gives that polarity essentially. And then we also have methyl groups. These are very prevalent in defining lipids and hydrophobic characteristics. So, and again, since it is hydrophobic, defines a hydrophobic characteristic of molecules, it gives that non-polar property. We also have carbonyl groups. Carbonyl groups are defined by a carbon double bonded to an oxygen, also giving that polar property. We have also carboxyl groups. These are very similar to carbonyl, but in addition to a carbon double bonded to an oxygen, you have a hydroxyl group there, as you can see. And you can identify these carboxyl groups to be acidic, so carboxylic acid. And again, it gives that property of acidity, definitely polar. And then we also have amino groups. These are nitrogen nitrogen bonded to two hydrogens. And these define molecules to be basic. We also have phosphate. Phosphate groups are defined by, again, as you can see, the molecular structure. Phosphate double bonded to oxygen, another oxygen bonded, and then as well as two hydroxyl groups. And then we have sulfohydryl, which is just a sulfur bonded to a hydrogen, also giving that polar property as well. All right, let me move my video in the middle there. Okay, cool. All right, so with carbohydrates, the main, I guess you could say, monomer, and you identify carbohydrates as, would be glucose. And as you can see, as I've written there, glucose would be like a monomer of certain carbohydrates. For example, sucrose is a combination of glucose and fructose. Fructose being a type of sugar as well, type of monomer. And then a single carbohydrate, the terminology of carbohydrates are kind of weird, but if you have a single carbohydrate, it would be monosaccharide, double would be disaccharide, triple trisaccharide. So I guess that is called Greek terminology, but mono, di, tri, also relate to that chemistry terminology if you name covalent compounds. But again, when you define carbohydrates, if you have one single one, it's mono, double, di, triple, tri. And then carbohydrates are formed through dehydration synthesis. This is actually very important, and it is huge in determining how these carbohydrates bond and how we can form not just a monomer, but again, for example, a disaccharide here or a trisaccharide. So dehydration synthesis is where the two monomers, for example, we could have two glucose molecules, as you see here in this diagram, and they bond, of course, like other monomers do, through a glycosidic bond, glycosidic linkage, forming H2O. What I mean by that is that after they bond, after these two monomers of glucose bond together, not only do we have this kind of, again, that this carbohydrate, this disaccharide, maltose, but we also have this result of water being formed. So you can see here that this, I guess, OH, this hydroxyl, bonds to this hydrogen. So you have two hydrogens and one oxygen, which results in water. So that's kind of cool. But just keep in mind that when you are defining a carbohydrate, when they bond together, dehydration synthesis is very prevalent. And the result of dehydration synthesis results in a water molecule as well, not just for example, not just a disaccharide or trisaccharide like this one, but again, water is also another byproduct. So now let's move on to lipids and phospholipids. All right, so lipids are defined by being hydrophobic. So as I mentioned, I think 2 slides ago with that methyl group, these lipids are really nonpolar. They don't like water. They don't interact with water at all. So again, they are very hydrophobic. Phospholipids, now these phospholipids are a special type of lipid molecule, lipid kind of structure, I should say, because they make up the plasma membrane of cells. They are arranged in a bilayer. So it's this structure here. This is the whole bilayer, but this is a single phospholipid. So kind of weird, but this head here, as we call it, is the hydrophilic part. And then this tail, these two, I guess, strings here are the hydrophobic sections. So why is the phospholipid bilayer organized like this? Why are the heads outside and why are the tails inside, facing inwards? Well, the reason for that is we know that, for example, water through osmosis passes through the cell membrane, right? So water interacts with cells. It has to. So that's why we need the heads to be facing outwards so that water can interact with the plasma membrane. But also, since the tails are inside, carbon dioxide, for example, which is nonpolar, can also pass through. These tails allow for nonpolar molecules to pass through as well, essentially. So keep that in mind. The heads are outward for water to interact with the cell. The tails are inside so that nonpolar molecules can also pass through the cell as well through simple diffusion. And then I also kind of included a unique diagram here. Phospholipid is this structure, triglyceride is another type of lipid. And then we have a steroid, which is, for example, cholesterol. But again, the main lipids you need to know are just the phospholipids and then the original characteristics of lipids in general. All right, so moving forward in terms of lipids and phospholipids, I also wanted to discuss what are called saturated and unsaturated fats. So with saturated fats, you can recognize them in terms of you have a chain of carbons essentially with hydrogens. But if you see compared to the saturated and the unsaturated has a double bonded carbon to, again, another carbon. And it's that double bond that makes the fat unsaturated. And saturated is just, for example, saturated fats are butter and solid forms of fats, essentially. Unsaturated are just liquid fats, for example, oil. But keep in mind that the double bond does not make the fat stable. That's why you could recognize them in terms of being liquid in a liquid state. But saturated fats, you can see that this carbon chain is subtle. It's not like this carbon chain here where this one is just leveling off a bit. It's totally stable. So that's why we recognize saturated fats in a solid state. So keep that in mind. And then this shows kind of the phospholipid, the plasma membrane, which is where the heads are. You can see here the phospholipid bilayer and the tails are inward. And then these are like some proteins, like peripheral proteins and stuff like that. But keep in mind, I just wanted to keep a diagram, show you guys another diagram of how the structure, then the phospholipid structure is organized. All right. Now, lastly, I want to discuss nucleic acids. I'm going to be very short in terms of discussing this section because I made a comprehensive video on nucleic acids alone, which was my very first video. But really, what you need to know about nucleic acids are the monomers, which are nucleotides. So the monomers are the nucleotides, and the end products are nucleic acids. So the end products could be DNA, RNA, and the bonds are pretty unique. The name, I should say, is pretty unique. Phosphodiester bonds. Pretty long name, but since it was kind of unique, it kind of stuck with me. But just the bond, that's the name. If you're studying for vocabulary, it's called phosphodiester bond. And I included a diagram. Of course, you have DNA, you have RNA. RNA is single-stranded. It is a type of nucleic acid. DNA double-stranded nucleic acid again. And then I included a simple diagram here showing the nucleotide, phosphate group, the carbon sugar, as well as nitrogenous base, which could be adenine, guanine, thymine, uracil, depending on if you're examining DNA or RNA. But quick recap here with the nucleic acids. Nucleotides are the monomers. The end products are nucleic acids like DNA and RNA, deoxyribonucleic acid, ribonucleic acid. And then the bonds are phosphodiester bonds. So keep those in mind. And I think with nucleic acids, you should be solid in terms of what you should know. But also my very first video, I should say, there's just a more comprehensive review on nucleic acids. And if you want more information, that you can check that out, as always. All right. Okay. Well, that concludes this part of the video. I hope you guys enjoyed it. Again, thank you so much, guys, for watching this part of the video. I hope the two-part lecture was a wholesome review of chemistry of life. If you guys have any other questions, of course, feel free to contact me, post some questions down in the comments, and I'll be more than happy to help you guys out. But until then, I will see you in a future video. Thank you so much.